So guys, Sriram Biology here from A Express, back with another biology video. Today we're going to be doing something a little bit different. Instead of doing a concept video, I'm going to be solving ATP video instead, as it's coming up soon. So without further ado, let's get straight into it. Let's just scroll down here. Okay, so, and also forgive me for my audio, it's raining, it's thunderstorm, so it's an entire thing. So first, the first question, always we have that experiment and the table questions coming up. So let's just read through this. A student investigated the effect of acid concentration on the rate of diffusion in agar jelly. So think of this. And the student was given 3 test tubes containing bromomethyl blue indicator in agar jelly. And the important sentence is, it is a blue indicator that turns yellow in the presence of an acid. So that's something we need to remember. It's going to be important. So let's just keep going down. The student used this method, labeled 3 beakers A1, A2 and A3. So this information, you might want to remember it, but don't give too much importance to it. This is what we need to know right for this question, because here you can see they've left A2 blank and they're asking us to write the final volume of the solution in beaker A2. So we'll get that information right here. So that you can see they're first adding 18 centimeter cubed of distilled water, and then they're transferring 2 centimeter cubed, no, that's from A2 to A3, sorry. They're adding 2 centimeter cubed into beaker A2, and then 18 centimeter cube of distilled water. So from this we can just figure out 18 plus 2, that is, sorry, 20. Oh, my stylus is not writing properly for some reason. That is 20, you know what, might as well just write with my hand at that point. That is 20. Okay, so let's keep going. So we did that. Now let's just read the rest of the experiment. They've added all these volumes and they have 3 beakers. So what they're doing now is they're labeling 3 test tubes with bromomethyl blue indicator in agar jelly. And use a cinch. Okay, so they're transferring volumes from these beakers to these test tubes, and it's in a water bath. They're starting a stop clock, etc., etc. So now let's get to the question. Here we have our tables. Okay, so they're asking us to measure the height of the yellow agar jelly in each test tube shown in figure 1.1. This is an important point, and prepare a table and record the results. So here we had to include the measurements and the final concentration of hydrochloric acid. So the final concentration of hydrochloric acid is actually already given here, so that we don't need to mind. We just need to copy it from here. What we need to do, you're going to have to measure the height here. But the thing is, obviously, because I'm doing it on my laptop, I actually can't measure it myself. I could just measure it on the screen, but that's going to be an inaccurate value. So when you're doing it, you can actually measure it and write your values. But I'm just going to write a random value for now, but I'm just giving you a disclaimer. So how we're going to draw this table now is, they mentioned the measurements, the final concentration. So that's like only 2 columns. So we can go ahead and draw a 2-column table. I know, please forgive me for my terrible handwriting. So this will be final, you know what, let me make that a bit better. So this 1 column is going to be the final concentration. So it's not a bit big because we have a lot of space, might as well. So first, I just prefer to do this one thing that when I was writing my boards, I would also write the test tube name just for safety. So let's make 1 column for test tube. Let's extend this line here. 1 column for test tube, 1 column for the final concentration, and another column for our height of the agar jelly. So just bring that down. Draw the table, top here for our labels. So let me just write that. Let's see if I can write it somewhat legibly. I just tell you what this is supposed to be. Test tube. Okay, that is supposed to be test tube. This is supposed to be final concentration of hydrochloric acid is a bit big, so I'll just write concentration, the T, and I, and finally again this is height of agar jelly, but I can't write that, I don't have enough space. So height And the unit is very important. So here our unit is mole per decimeter cubed. So again, I'm just going to write this in short form. So instead of writing per decimeter cube and everything, I'm just going to write dm power minus 3 because that's essentially per decimeter cube. Please keep in mind, guys, I'm just taking some shortcuts here because of the technical difficulties. Please don't actually do this. Mole per dm power minus 3 mole per decimeter cube, and for the height it's just millimeter. Okay, so now we have 3 test tubes, so we divide that into 3 rows. So A1, A2, and A3. Okay, so the concentration, we have it right here, is 1.1 and 0.01. So just write that out. God, the table is rather horrible to look at. Please forgive me for that. 1.1 and 0.01. So like that. Okay, and the height, as I mentioned, I'm just gonna write a random value because I don't actually have the printout with me and I can't do that off screen, so I'll just write a random value. So it's in millimeters, keep that in mind. When you're doing the paper, please measure it accurately. So I'm just gonna randomly write 25 millimeter, 14, and 5. So I'm just gonna write those values, but actually please measure it accurately and write it in millimeters when you're doing it. So that's a table. So we have the test tube column, a concentration column, and a height column. So these are important details, and we've scored 5 marks already. Now we go down. State a conclusion. So the theory that what they're trying to investigate is the rate of diffusion of agar jelly. So in the agar jelly. So what we can see here is it turns yellow in the presence of an acid. So the yellow agar jelly is essentially agar jelly that contains acid. And when it's blue, that's like when there is no acid. So as we can see here, the concentration of acid is higher. Like A1 has the highest concentration of acid. It has the highest height of agar jelly, that is yellow. It has the most agar jelly that contains acid. So we can say that when there is an increase in the concentration of the hydrochloric acid, there's an increase in the rate of diffusion, because we can clearly see that relation here. When there's going to be more diffusion, so more acid is going to be entering the agar jelly, so higher height of yellow agar jelly. So let's just, that was an accident. Okay, let's just write that as our answer now. Slow down. So, okay, let's try writing this. Increase in concentration. Here I'm just going to write a short form. Please do not do this in your exam. Increase in concentration of acid increases rate of diffusion. Okay, so increase in concentration of acid increases rate of diffusion, and the independent variable and dependent variable is rather obvious. So the independent variable is the concentration, because that's what we're varying, like we're trying to vary, and the dependent is the height of agar jelly, but again due to constraints I'm just going to write the short form. Please guys, I'm going to repeat this time and time again through this video. Do not just write the short forms like I am. I'm just writing it for convenience, because writing out the larger words just makes it more illegible. So I'm just going to write concentration is our independent variable, and of acid you can just write that. Dependent variable is height of the yellow agar. Okay, let's move on. So these are the easiest 1 mark questions you can do, like this temperature one right here. So first, be careful about the scale. So here we can see it's 20 to 30 in 10 lines. So each line is 1 degree Celsius. So 21, 22. It's on the third line. So that's going to be 23 degrees Celsius. These are all easy ones. So students stated that the change in temperature of the water bath was not a significant source of error. So actually the water bath was supposed to be, I think 35 degrees Celsius. But now when they're checking it, it's 23 degrees Celsius. So why is this not a significant source of error? So what we have to realize is all 3 test tubes are in the same water bath. So a change in temperature is actually going to affect all 3 test tubes the same way. So that's why he's actually correct to be stating that. Let's say each of them are in a different heating bath or something, then one of them being a different temperature obviously would be a significant problem. But here, where we have all 3 test tubes in the same water bath, having a change in temperature is not actually going to affect it much. So let's just write that. So and the change in and yes, I'm going to be almost, no, I did not cross the margin. The change in temperature affects all 3 test tubes equally. Suggest a control experiment. So control experiment is only the null hypothesis. So we're not actually going to have like acid or whatever. We're just going to have like a normal result to compare our other test results. So what we can do is we don't want like the independent variable to be changed. Like we don't want to be changing the concentration of acid or anything like that. So maybe instead of using acid, we can use like water or something like that. That's a good control experiment where we don't want to actually cause any variation in our results. We just want something to compare with our actual test. So here we can just put something like use water instead of acid. So we can write something like that. Because again, control experiment is something where you just want a basic result to compare with your actual tests. You don't want to vary anything like the independent variables, just a normal null hypothesis. So the student repeated the investigation but left the test tubes in the water bath for 30 minutes instead of 20 minutes. Okay, so here this would actually increase the height of yellow agar, because again this is going to increase the rate of diffusion. So you're heating it for longer, it's going to diffuse for longer, and it's going to increase the height of agar jelly in all 3 test tubes because they're going to interact with the acid more. So here again, let's just write that we're just going to write that the height of the yellow agar jelly is going to increase for all 3 test tubes because the rate of diffusion will increase. But they're just saying suggest how to differ. We don't need to explain why. Suggest. Keywords are really important in the questions. So suggest means we just need to say how it might change. So height, and we can't just say it's going to change. We have to say like in which way, like decrease, increase. Here it's going to increase. Height of yellow agar will increase my handwriting is horrible. Increase in all 3 test tubes. Okay, so now we are reaching the next part. In an experiment, a student cut different size cubes of agar containing bromothymol blue indicator. The student places the cubes in hydrochloric acid and measures the time taken. So here we can say just from this question, we can see different size cubes. He's cutting different size cubes. That is going to be the independent variable, the length of the cube size. You're going to understand why I'm saying this very soon, but I'm just going to differentiate. So the length of the cube size is what's going to be our independent variable, and the time taken is going to be our independent variable. Just keep that in mind. So results are shown. Okay, obviously they've left it blank. They want to solve something there. So complete table 1.2 by calculating the volume of the 4mm cube. So there are two very obvious. A, you might already know the volume of the cube because of math. It's just a cubed, a being the length of the side. So this is going to be 4 cubed, which is 64. Or you can see very obviously, if you forgot for example, like you blanked out, you can see the unit for length is just millimeter, but the volume is millimeter cubed, and they haven't given any other side, so it's just going to be the same thing cubed. So from that itself, I can tell that this is just 4 cubed, which is 64. Let me just write that. Now I think it's a table we have to draw next, a graph, sorry. So yeah, plot a line graph on the grid of the length of the cube sides, which is our independent variable. Now you're gonna understand why I mentioned that, against the time taken. So whatever they say first is the variable you need to put on the x-axis, and whatever they say after the word against is what you need to put on your y-axis. So here, length of the cube sides is gonna go on to the x-axis, and the time taken for the cube to become yellow is just going to be on the y-axis. So first we need to label the axis with our units. We need to give it a scale. So length of the cube sides, it's in millimeter like they mentioned. I am not going to write out the entire name, guys. You don't need to in the exam either, but just to be safe, write out the entire variable, like length of the cube sides. For me, again, because of my technical difficulty, I'm just going to write length and leave it at that. Length, and I'm just going to write time taken. They took it in seconds, so we also take it in seconds. So time taken in seconds. So we have our axis labeled with units. Now we need to give it a good scale, and always, guys, just to be safe, take a scale that will fit 3/4 of the axis. Of the ms, each ms it changes. Some say half, some say 3/4. But to be safe, always try to take an axis that fills at least 3/4 of the grid in both directions. So here, the length of the cube sides, we can just take it as 2, 4, 6, 8, 10. Like it's increasing evenly. And if you 2, 4, 6, 8, 10, it fits perfectly. Let's just write that right here. So 2, 4, 6, 8, 10. So that's perfect. Now the time, oh god, I keep doing that, sorry. The time, it's going from a small value 35 all the way up till 445. So here I think the best scale, because we have 5 boxes, we take 100, 200, 300, 400, 500. That's a perfect axis that we have. So we can start from 100, 200, 300, 400, and finally 500. Just, yeah, 300, only 500. So now we're going to plot our points, and how we're going to do that is we're going to draw a small dot and then a circle around it. That's how they prefer it to be drawn. So first is the length is 2, and the corresponding time is 35 seconds. So here we're not going to have the exact thing like 35 or something like that. It's going to be a bit in between. So each small square is 10 seconds on our y-axis. So 2 is 35. So the first line is 10, 20, 30. So it's going to be like in between the third and the fourth. So I'm just going to try plotting that as accurately as possible. 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. Okay. 10, 20, 30. That's about right there. And then we draw a little circle around it like that. Not that big of a circle, but a decently sized circle around our point, and then our next point 60. This is going to fall exactly. So length of the cube side is 40 and 60. 4 and 60. So 4 millimeter here. So 60 is right there on. I can't get it quite on because of the sensitivity of the screen. Just yeah, right about there, and we draw a little circle around it. If you see, like, I'm trying to get it exactly on the line, but because of the sensitivity of my screen, I can't quite do so. Let me just retry that. Sorry. Seems like the screen wants to touch the stylus is touching every part but that point. It's getting kind of annoying. Yeah, okay, that third dot that we have, we're just going to draw a little. Okay, then we have 6 and 170, so it's a big jump. 6 millimeters, we're going all the way up to 170. So 170 is just 3 squares below 200. So 190, 170, which is perfect. Finally we got, oh sorry, that was a huge circle. Let me just trim that down a little bit. Let me just draw a little circle around it like that. Perfect. Then we have the length is 8 millimeters and 275. Again, this is going to be in between two squares, and it's going to be very annoying for me. Okay, so 8, 275, 60, 70. It's going to be in between. Let me just try getting this. No, a little bit below. There you go. So that third little dot that I've drawn on the bottom left of those 3 dots, that's 275. Finally 445. So 10 millimeters, 445 again. Now we're going up between 440 and 450, the halfway line. So we get 10 units, and out of that between the 4th and 5th. So 10, 20, 30, 50, just somewhere. That was very inaccurate. Yeah, it's good enough. Wait. 1, 2, 3, 4, 5, 6. Yeah, it's correct. Okay, so now here they've said a line graph. So the important thing you need to know for biology is you almost always never have to draw a best fit line, which is connecting most of the points. Like, you almost always never have to draw one in biology unless they mention it. A line graph is where you're going to connect each point. So you're connecting each point to each other by a straight line. So obviously my lines are not going to be too straight, they're not going to be too accurate, but I'm going to try to draw it as good as I can. But you're going to, when they mention line graph, you're going to have to draw it by connecting each point to each other. So let me just do that for you right now. So you're going to want to connect each point. So that's like that. That was supposed to be in the middle. No, okay, no, that's correct. Then draw like that. Then like that. Finally, in halfway, like that. So you're going to redraw the same thing, but with straight lines and obviously way neater than what I've done. So now, okay, so what they've asked is find the side length of 3 millimeters to become yellow. The time taken for a cube with the side length of 3 millimeters become yellow. So 3 is halfway in between 2 and 4. It's right about here. So with your graphs, you're going to get a much neater diagram and a much more accurate diagram in which you can take better readings. For me, it's that 3 millimeters touching the axis right about there. I just take it like this. That's about 50 seconds, I think. Yeah, that's about 50 seconds. And for me, but for you, it's going to be a much more different value when you draw an actual proper accurate graph. So what is it? So please don't draw a best fit line unless explicitly mentioned. Here we just connect each point with the line in between, straight lines. So 2 variables that should be kept constant. So just think here, what we're varying is the length and the time. Length of cube sides and time, those are independent variables. So if you want the results to be consistent and you don't want them to be different, too different between each result, you only want the length of the cube side to affect the result, then you should keep, there are so many things you can keep constant. You can say temperature, you can say the concentration of hydrochloric acid we're using, you can say the volume of acid, the pH of acid. The first two things that came to my mind when I was saying it was the temperature and the concentration of acid. So I'm just going to go ahead and write exactly that. So temperature could be any temperature. You don't have to specify that. And concentration of acid. Keep on moving. We're going faster. Okay, so now we've reached the second question. We're finally done with those agar experiments. Now we have a drawing to do here. So for drawing, I'll just give you some general tips. So always for drawing, try to draw 2 times the size of the drawing they've given. So that AB line, you measure it and you redraw it, and make sure when you draw a line from the same points in your drawing, it should be, example, let's say this is 2 centimeters. When you measure the same points in your drawing, it should be at least 4 centimeters gap between them. You want to magnify your image by 2. You want to draw 2 times as big as the image they've given. Again, because I don't have this on paper, my measurements are going to be inaccurate, but I can roughly see how big it is. I'm just going to try drawing it 2 times bigger than this. So the thing is, you don't need to be like Picasso or like an amazing artist. You just need to get the important features right and make sure make it obvious what you have drawn. So the details, we need to take into the key details and show that in our drawing as well. So let's say here we have the radical, which is what's going down. This is the radical, what's going downwards here. It's forming the root, and you have the plumule, which is forming our shoot. So we can see that our radical is much bigger than our plumule. It's almost 2/3 bigger. So we don't need to maintain that exact ratio or anything, but you need to show obviously that evidently that it's bigger, and you need to get like the details, like this part, you need to get this right, and you need to show the way that it's curving. You need to get all that right, and you do not want a broken outline. If your outline isn't broken or it's not smooth as it has to be, erase it, redraw your drawing. No way around it. I am probably going to be redrawing it multiple times because my drawings are quite bad, but I still managed to score pretty well in my exams because I would just follow what my teacher said, which is outline always consistent and smooth. It's broken, redraw it, get your key details right. That's all you need to know. So let's just zoom out a little bit so it's a bit easier for me to draw. Okay, so let's start like this. It's going to be much bigger. Let's start like this. Okay, see, for me, when it's breaking, it's because the stylus is not very accurate. It's going to lose contact with the screen. That's why it's going to break. So I might not be redrawing it that many times. Only if I genuinely mess up with the outline, I'll redraw it. But so please guys, when you're drawing with the pencil, ensure it's very consistent. For me, it's going to keep breaking because of my inaccuracy in the technology that I'm using. So please be careful when you're doing it. So let's start with the underside. You come up, you curve around, you go down, and then you have the plumule. It's going to go, let's go scroll down a little bit. Scroll down. It's the plumule, it keeps going down, comes back up. So you see, obviously this drawing is really bad. Like, it's not nearly as good as their drawing, but you see the key details. This circular section, we got it right. Then the plumule being much bigger than the shoot. The radical being much bigger than the plumule, we got that right as well. You can see it's like much bigger than the plumule, so we got that proportion right. And the circular section, we don't need to get the extreme fine details right or anything like that. So this is our drawing. Obviously, when you're drawing in your pencil, please make it much neater than I have, and I'm confident that you will do that. So let's just leave our drawing there. That drawing is not very good looking, is it? Okay, so measure the length of line AB. So when you are measuring it on paper, it is definitely going to be way more accurate for you than it is going to be for me when measuring it on a screen. But just for the sake of this, I am going to take the measurement of the line from the MS just to ensure it's accurate enough for you. And so please ensure that you are measuring it accurately and you're doing it on paper. I'll just take the measurement from the MS. It is what the value you're supposed to get when you're measuring it on paper is supposed to be 31 millimeters or 3.1 centimeters, but we had to write it in terms of millimeters. So you should be getting 31 millimeters. Plus or minus 1 millimeter is fine, but ensure you're getting something around that line. So now IGCSE is kind enough to just give us the formula for magnification and the values. So this is an easy 3 marks to score here. It's fine. It's very easy. Just follow the instructions as they're given. So actual width of the seedling is 9 millimeters. Length of line AB which you measure is 31 millimeters. So simply for magnification they're given length of line AB by actual width. So that's just going to be 31 by 9. You run this in your calculator. It should be around somewhere like 3.4, I think. 3.44. So that's 3.44. But 2 significant figures. So 3.44 is 3 significant figures. We're just going to round that down and write 3.4. We're done there. Let's keep on going. So we're done. Easy 8 marks. Here, just please remember, don't give too much importance to the details and just don't give too much importance to the very extremely fine details. Like, don't try to make it seem pretty or seem good or seem extremely accurate. They just want essentially an outline of what they have given, because we don't need to get the little example you see on the plumule. You have those little strand things. They don't want us to represent all that. They just want us to get the external details right. That circular tester, then we have the larger radical and the smaller plumule. We just need to get these kind of details right. Wait, actually no, I'm sorry. The details like the leaves, actually these little leaves on the plumule, we actually have to get those details right. I am very sorry, I kind of didn't consider that. I think if I erase this, it's going to erase the entire thing, isn't it? Yeah, it is. It's fine. I'm sorry. Let's just redraw that. That is an error on my part. Very sorry. So when we are drawing it, we need to get the external details right. We don't need it to look pretty. We just need to get the external details. So these 2 leaves, I actually forgot to consider them when I was drawing it. Sorry, very, very sorry about that. Please remember to draw it when you are doing such things. So let's just, I don't know what's happening there. It's like a glitch. Okay, so just zoom out. Okay, so we're gonna start again, again, when you're drawing it, don't let that be a broken outline. For me, it's just because it's stylus. Go up, go down, small leaf-like part here, and then you have the small plumule. Again, the outerwards curved leaf. Again, when you're drawing it, don't let the outline become broken like this. Just let that go quite a bit down. So see again here, the radical much larger than the plumule. We've shown 2 outerwards curved leaves on the plumule, and we have mentioned those we have drawn those details. Again, when you're drawing it, do not let always a common point that you get 1 mark is a clear unbroken outline. That's any MS you check, that's going to be a point. Here, because of my stylus, the outline's a bit broken. When you're drawing it, do not let the outline get broken. Ensure a consistent smooth outline. So sorry again for the mistake of not drawing the leaves. Please remember to. So let's now zoom back in as we move on to the next question. Okay, so figure 2.2 shows photographs with 2 different seedlings P and Q. Describe 1 visible similarity and 2 visible differences between seedling P and seedling Q. So the first thing I see here is both have a radical present. That's the first thing I can think of, because we can't say they both have a plumule present, because here we can evidently see the stem, the plumule growing, whereas here we only see a radical, but in both there is a radical. So I'm just gonna write that immediately, the most obvious thing. You can also say there are root hairs present on both, because I can see the little root hairs present in both. And so those are 2 ones. And you can also say the seed covering or the tester is broken in both. We can evidently see here It's cracked here also, it's evidently cracked, but I'm just going to go with that point that radical is present in both. Radical present. And the difference that we have here is P has a plumule, whereas Q doesn't, but we don't need to write in like P has plumule, Q doesn't. We can just write P has a plumule. And the another one is the root hairs on Q are fibrous. So we can write it like in terms of Q here. We can just write Q has fibrous root hairs. So that's the first thing I thought of, because you can evidently see here it's just like little little deviations from the root. Here it's like a fiber, it's like hairy looking. So that's like a very obvious thing we can write. Now this is something you're gonna have to learn, you're gonna have to remember. So as seedlings grow, they change starch in reducing sugars. So describe the method you would use to test for starch and reducing sugar. So this you're gonna have to remember. Starch is the iodine solution test. We don't need to give the result because they haven't mentioned us asking for the result. In iodine is blue black, reducing sugar it's I think brick red. So starch test, just describe the method. We just use iodine solution. You don't need to heat it or anything, so you don't need to mention that. Just write use iodine solution. Key terms are very important, guys. Please check the key terms for each question. Then reducing sugar test, we add Benedict solution and we just heat it in a hot water bath. But we just need to write use Benedict's solution and then heat. So that's our points that we have there. Now we're done with that. Now we're moving on to the last question of this paper. Yep, that's the last question of this paper, the experiment. So when light shines from one side, seedling grows towards the light. It shows the diagram. Okay, so here the dependent variable is the angle at which the seedling grows. So here we need to show the 6-mark experiment. Seems like a huge task to do, but you just actually had to break it down into its parts. So you need to write about the variables you need to keep constant. You need to identify the independent variable. You need to give a procedure. You need to mention something about accuracy, and you need to mention something about safety. That is literally it. So here you don't need to mention the dependent variable because they've already given affects the angle at which the seedling grows. That is our dependent variable. That's what's going to be changing. How light intensity affects. That phrase tells us that light intensity is the independent variable. So we had to identify that. That is 1 mark already in the bag. So 1 mark is for the IV. Okay. And then you can, another 5 marks are left. So 1 mark is for the accuracy. 1 mark for accuracy. So that's like a point mentioning like repeat the experiment and take averages, something like that. 1 mark for safety. That leaves 3 marks for us left to score. So in this, you need to mention the procedure again. So you can break it up in different ways. You can either mention you can say 2 marks for constant variables and like 1 mark for the procedure. But I don't like doing that. I usually like actually going a bit more than the total. So even though there's only 3 marks left to score, I am going to mention 2 constant variables and 2 marks for the procedure. 2 marks for constant variables is going to give 2 constant variables are going to give me 2 marks. 2 marks for procedure are also going to give me 2 marks. So 2 plus 2 plus 3, that's 7. But it doesn't matter. We need to score all 6. So let's be safe and just write everything. So first, the independent variable. I'm just going to write it in abbreviations. Guys, please, 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 I'm begging you, do not write abbreviations when you're in your board exam or whatever ATP paper you're doing. So the independent variable, use minimum 2 different light intensities. That is our independent variable, light intensity. Minimum 2 different light intensities. Also guys, try, obviously here I'm writing humongous letters. When you're writing, try not to go over the lines. If you do maybe 1 or 2 lines, you don't want to give a bad impression to the examiners. So use minimum 2 different light intensities. Now we have constant variables. So here we can mention like something like, well, first let me write something, then I can think of something. You're going to want to plan it out before you write it. Constant variables. So here it's plants, and so we might want to take something like the type of plant, because the type of plant can definitely change the way it grows, etc. So type of plant is one constant variable, and also the time. The time is also an important variable, I think, because the time is going to affect how much it grows, and how much time it's under the light intensity will affect the angle. So let's just write type of plant and also time. Okay, so now I'm going to go over the lines. Please guys, don't go over the lines when you are doing this. It's type of plant and time. Now proceed. I'm just going to write it here. Procedure. Procedure. So the procedure, we are changing the light intensity, but how? We need to mention how. So let's say, like, let's use different lamp distances or different bulbs. I think I'm just going to say use different light bulbs to alter the light intensity. And then you have to measure, say, a method of how we're going to measure the angle. So let's say use a protractor. So something like that. So first, differ light intensity by using different light bulbs, and then measure angle of what? Angle of the seedling. We can't just say angle. Measure angle of seedling using protractor. Oh god, almost out of space. So for the accuracy, repeat the experiment at maybe 2-3 more times. So let's just say a point of the accuracy here. Repeat experiment. You can write any number. I'm just gonna write 2 times. 2 more times. At a point for the safety. So let's see what we can write about safety. Actually, this one's a bit tricky to think about something for safety, because here it doesn't act, it's not very dangerous or anything. But I guess we can use a heat shield, because it's going to be quite hot when you use higher light intensities. So use heat shield. Use heat shield for safety. Okay, and there you have it, guys. That's the entire May June 2024 6-2 paper solved. I will be adding chapters so you can see which part of the paper I'm solving in the video. And also please pay attention to the error I made. In the drawing section, I corrected it later on. And yes, please, thanks for watching the video, and please put in the comments which paper you'd like to see us solve next. Thank you. Bye-bye.